Hello there, vetmates! Today, our group consisting of Michael John Iblugin, Eric John Perono, Cristal Marie Mendoza, Ella Faith Tacardon, Gwyneth Colleen Lopez, Ariane Clarice Bautista, Helen May Barsubia, Alana Nicole Casilak, and Ivan J. Guleng is diving into the world of aquaculture and aquatic animal management. Let's learn and explore together.
Immunity and biological methods of disease prevention and control. The immune systems of fish and crustaceans have similarities and differences. Fish immune system has these non-specific mechanisms of defense, but they can be differentiated from those of crustaceans, as they have evolved an additional way of recognizing microbes. This recognition is the basis of what is commonly called the adaptive immune system or specific immunity. The adaptive immune system has two hallmarks that distinguish it from nonspecific immunity. Firstly, recognition is performed by a receptor that exists in billions of different forms in an individual. And secondly, the adaptive system retains a memory of each particular microorganism to which it has been exposed. The receptor that is responsible for these remarkable properties is the antigen receptor. Immunity in fish is mediated by two major systems, the innate immune system and the adaptive immune system. The innate immune system is thought to be an ancient origin and acts as the first line of defense against invading pathogens. On the other hand, the adaptive immune system is present only in the vertebrates above the level of the agnatans. So we have here the table, the major components of the immune response in fish. First, the soluble factors. Innate immune response, or acute phase proteins, enzyme inhibitors, cell lytic enzymes, and agglutinins. For adaptive immune response, we have the antibodies. For the cells, the innate immune response is phagocytes, natural killer cells. And for the adaptive immune response, the B lymphocytes and the T lymphocytes. Innate or nonspecific immunity refers to various physical and cellular attributes that collectively represent the fish's first lines of defense against infectious diseases. First, we have the physical barrier. The skin, including the scales in some fishes, and the mucous membranes of the digestive tract are physical barriers that protect the animal from harmful environmental agents and from invasion by microbes. Most of the fluids that are excreted or secreted onto epithelial surfaces, such as the mucose and digestive juices, contain chemical components that are antimicrobial. Mucose is secreted by specialized goblet cells in the epidermis of fish and contains immunoglobulins. Mucose also contains precipitins, natural agglutinins, lysines, lysozymes, C-reactive protein, and complement. We also have here the soluble humoral barriers. The internal fluids of fish contain a number of substances that react with a variety of microbes to lyse, or coat them, or to inhibit their growth. So we have here 7 soluble barriers. First, we have the acute phase proteins and complement factors. The concentration of a number of protein types in the blood serum increases rapidly during an infection. So these proteins are called acute phase proteins. On the other hand, the complement factors of fish blood are also proteins. Second, we have the transferrins and lactoferrin. Transferrin plays a possible role in delaying the start of microbial infections by binding the available iron and thereby depriving bacteria of an essential growth factor. On the other hand, lactoferrin is the mucous secretion of mammals, contain another closely related iron-binding and antimicrobial protein. The third one, we have the ceruloplasmins. So ceruloplasmins acts as an oxidase that oxidizes divalent iron to ferric ions, which then bind to transferrin. The fourth one, we have the metallothionein. It is a peptide rich in cysteine and with a high affinity for metal ions. The fifth one, we have the enzyme inhibitors. So these are present in fish and are believed to play a role in retarding the invasion of pathogens or parasites. Next one, we have the cell lytic enzymes. Lysozyme is present in most tissues and secretions of fish, and the level seems to vary in relation to environmental conditions. And lastly, we have the agglutinins. So these agglutinins are nonspecific, and they correspond to lectins of invertebrates in their ability to recognize and bind to single sugars on the bacterial surface and mediate phagocytic reactions. Lastly, we have the cellular barriers of the innate immune system. Under these, we have the inflammation. Inflammation is the observable condition that accompanies damage to the body. It is a localized response to tissue injury and to invading microorganisms. It is characterized by an infiltration by granulocytes and macrophages, removal of dead cells and foreign cells and debris, followed by tissue repair. Inflammation confers protection by walling off uninfected area from the rest of the body. Second, we have the natural cytotoxic cells. The non-specific cytotoxic cells in fish are equivalent to the natural killer cells of mammals. They differ from their mammalian counterparts by being able to destroy a wider range of foreign cells, and they can even destroy multicellular parasites that attack fish. Lastly, we have the phagocytosis in fish. Macrophages, monocytes, and granulocytes are phagocytic, and in some species, neutrophils are also phagocytic. Activated macrophages produce oxygen metabolites, such as superoxide anion, in a process known as the respiratory burst. So let's move on to adaptive or specific immunity. Under adaptive immunity is we have the antigens. Most antigenic substances are large molecules like proteins, polysaccharides, and nucleic acids, but usually the immune system only recognizes and responds to a small part of these large molecules, called the antigenic determinant or the haptin. The second one is the cells. Two types of cell that are involved in specific immune responses are lymphocytes, the B and T lymphocytes, and also the antigen-presenting cells, which includes the macrophages. Lastly, we have the receptors. Lymphocytes have membrane receptors for specific antigens. These receptors are proteins that specifically recognize and binds to an antigen. They are the means all lymphocytes use to recognize antigens and some use to respond to them. We also have here humoral and cellular immunity. Humoral immunity acts by the secretion of soluble proteins, such as the antibodies or immunoglobulins, that circulate in the blood and lymph where they can combine with antigens and neutralize them. Humoral immunity can be either active or passive. Active immunity occurs when the antibodies are made within the body's immune system. It is passive when the antibodies come from outside the body. The next one is the antibody production. Plasma cells are the effector cells of humoral immunity. Plasma cells are B lymphocytes that have been activated by interaction with their matching antigen. Besides the B lymphocytes themselves, it involves accessory cells such as macrophages and a specific class of T lymphocytes, which are the helper cells. All cells except RBCs have MH1 proteins, and only a few types of cells have MH2 proteins. This includes macrophages and other accessory cells that can function as antigen-presenting cells. Next, we have the immunologic memory. Immunologic memory has primary and secondary responses. So the first time the body is exposed to a particular antigen, the resulting immune response is mild and brief. But the second time that the same antigen is encountered, the response is strong and long-lasting. So aside from that, we also have the memory B and T lymphocytes. So activated B cells that are formed as part of the primary immune response become short-lived plasma cells that produce antibodies. Others persist as memory B lymphocytes. So these cells are able to multiply and transform rapidly into plasma cells during a secondary response, when they produce some IgM and lots of IgG. In the activation of T lymphocytes, memory T cells are also formed. Lastly, we have the cellular immunity. Cellular immunity acts by direct cell-to-cell contact to protect the body against viruses that have infected its own cells and against tumor cells. T cells have membrane proteins that bind only to a specific antigen, just as B cells do. T cells rely on displaying fragments of antigens combined with MHC proteins. Cytotoxic T cells are the effector cells of cellular immunity, and they also attack foreign cells that are present in blood transfusion and tissue transplants. Crustaceans' immune system. Crustaceans are no different from other animals in that their defense system is largely based on the activities of the blood cells or hemocytes. The crustaceans' hemocytes carry out phagocytosis, encapsulation, produce antimicrobial substances to remove or neutralize foreign particles and infectious agents. Another hallmark in crustacean immunity is the rapid sealing of wounds by blood coagulation to prevent loss of hemolymph and to immediately entrap invading microorganisms and arrest their dispersal in the body. Based on their morphological appearance and their staining properties, different hemocyte types can be distinguished in crustaceans' blood. However, these hemocyte types do not fall into the same groups as those found in fish and other vertebrates. The three types of hemocytes that can be isolated from crustaceans are the hyaline, semigranular, and granular. There are three defense mechanisms in crustaceans. First, we have the phagocytosis. In crustaceans, these are the hyaline hemocytes. Their primary function is to clear the body of foreign particles including virus, bacteria, and fungal cells. Second, we have the cytotoxicity. Warm-blooded animals, higher than vertebrates, produce a specialized white blood cell capable of killing tumor cells and cells infected by virus, and such cells are called the natural killer cells. And the last one is the lectins. Lectins are proteins or glycoproteins that have the ability to recognize and bind to the carbohydrates on the bacterial or fungal surfaces. Suppressor cells exist in the immune system to turn down antigen-driven responses and as a mechanism to maintain tolerance to self-antigens in the periphery. Thus, the immune system has a self-regulating mechanism to modulate its reaction, especially to cells and tissues of its own. However, there are many situations where the suppression of the immune system is unwanted and may eventually lead to disease. So there are a number of internal and external factors exist that can cause this suppression. First, we have distress. So nutritional stress due to improper diet, environmental stress due to poor water quality, physical stress due to handling, crowding, or confinement. We also have here the metals. So aluminum, arsenic, cadmium, chromium, copper, lead, mercury, nickel, and zinc are heavy metals that have been shown to lower non-specific and specific immune response in fish, making them more susceptible to various viral and bacterial diseases. The third one is the pesticides. The endrine, malathion, methyl bromide, trichlorphon, DDT, Belucide, and tributylene have immunosuppressive properties ranging from reduced lymphocyte number and phagocyte activity to necrosis of the thymus. Lastly, we have the drugs. Oxytetracycline is the antibiotic most used by fish culturists in treatment of bacterial disease. It has been long known to be immunosuppressive in fish, reducing the numbers of antibody-producing cells. So here are some of the biological control. First, the avoidance of pathogens. Under this is the pathogen-free water supply. A pathogen-free water supply is obtained through the sterilization of fresh water or seawater. First, we have the filtration. Ultrafiltration of water supplies through sterile of 0.2 micrometer membrane filters has the advantage of having none of the detrimental effects associated with the other methods of water sterilization. Second, we have the ultraviolet light. It reduces the reproductive capacity of bacteria and fungi and is therefore bacteriostatic or fungistatic rather than bactericidal. The third one is the ozone treatment. Ozone is highly oxidizing form of oxygen. Ozone will disinfect and sterilize seawater, oxidize organic material, and oxidize toxic nitrite to less toxic nitrate. Fourth is chlorination, less effective than ozonation, but is more popular form of seawater sterilization in aquaculture facilities in Southeast Asia. Chlorine, as calcium or sodium hypochlorite, is commonly used in hatcheries in South Asia. Next, we have the pathogen-free diets. In most cases, the preparation of commercial feeds involves high temperatures of about 100 degrees Celsius for short periods that should be enough to pasteurize if not sterilize the feed. On the other hand, the feeding of live food may present a different set of problems in that some germicides that are used to disinfect live feed could also be toxic to the live food organism. Bath treatments with a nitrofuran derivative, sodium nitrofurans, is live diets such as rotifers and brine shrimp are effective approaches to decreasing the number of bacteria in these diets. Lastly, we have the specific pathogen-free stocks. It is defined as animals that are free of specified microorganisms and parasites but not necessarily free of others. In prawn farming, some pathogens causing epizootics during the grow-out phase have been traced to be vertically transmitted from wild broodstock that are carriers of diseases. Although the use of SPF broodstock does not result in disease-resistant or even disease-tolerant stock, it is proving to be one effective managerial control measure, which minimizes the likelihood of epizootics due to an identifiable pathogen. Another component of biological control is the role of stress in disease. Aquaculture practices that increase stress are high stocking densities and poor water quality, injury during handling, improper nutrition, and poor sanitation. There are some managerial practices that may help prevent stress, and these are the fresh water quality, good handling and transporting, nutrition, and sanitation. So the next one is the improvements in host resistance. We have two. First, the dietary enhancement. So variety of nutritional components can influence the incidence and severity of a number of infectious diseases. Some micronutrients that are known to enhance disease resistance include vitamin C, B6, E, and A, and the minerals iron and fluoride. Second one is the vaccination. Vaccination in fish is possible, and specific vaccines can be developed along the same principles as for warm-blooded animals. So under vaccination, we have bacterial vaccines. So most of the vaccines are focused on salmonid pathogens, and a few are being directed against bacterial pathogens in carp and catfish. There are still no commercially available vaccines for bacterial diseases in warm water fish. Second one is the viral vaccines. The only commercially available vaccine against a fish virus was the spring viremia of carp, or the SVC, caused by Rhabdovirus carpio. It was administered by injection since the disease affects carp at a size and age of 9 to 12 months when they are easy to handle. The third one is the parasite vaccines. Vaccination techniques are being developed against parasitic protozoans by intraperitoneal injection of live attenuated parasites. Some evidence shows the passive transfer of protective immunity against these parasites from immune to naive fish and to egg. So there are types of vaccines. We have inactivated, live attenuated, recombinant, and DNA. We also have here the modes of vaccination delivery. First, direct immersion. Direct immersion is commonly practiced in very small fish. The second one is the spray administration. It is suitable for larger fish than those given immersion treatment. The third one is per oral administration. It is also suitable for larger fish than those given immersion treatment. And lastly, injection. This is the route of administration commonly used throughout the salmon industry. Our last biological control, we have the biological modification of the culture system, and under this we have the biological filtration. It facilitates the purification of the water in high density, semi-closed or closed aquaculture facilities by the oxidation of ammonia to nitrite and nitrite to nitrate. The second one is the probiotics. Probiotics are a group of food and feed products for human and animal consumption and are also known as direct-fed microbials. In aquaculture, it is commercially available probiotic products contain bacterial inocula not for consumption for fish but for the environment. The third one is the polyculture. It is a practice of keeping several of different species in the same pond. Polyculturing fish and prawns with filtering organisms to reduce phytoplankton, bacteria, and organic particles that had been done on the experimental scale. Oysters, clams, macroalgae, and bloodworms are often utilized to accomplish this cleanup. Lastly, we have the phage therapy. Phage therapy is the use of bacteriophage to treat bacterial disease. Phages are specific to their bacterial host, and since phages are specific, they cause much less damage to the normal microbial balance of the host.